All right, so here's when we are some overview. When we are trying to predict where are there genes in some genomic sequence, so think about, sometimes I have with me like a recipe book, and like when you buy a magazine for recipes, there would be some recipes and then some commercials in there, right? We could think about the genes are the recipes because they're instructions on how to make something. So the genes are the DNA that are instructions on how to make protein. And when you buy that magazine, there will be some recipes and some advertisements or whatever else. So my analogies here is that the genome has genes that encode proteins or genes that encode RNA molecules that are functional as RNAs. So those are referred to as RNA genes. And then in case of eukaryotes, there's a lot more DNA that doesn't actually encode anything. So in our cells, you could think that we are magazines that have recipes, but they have many, many more advertisements than recipes. So that would be the non-gene DNA, not only non-coding, but non-gene. So the word coding is for this DNA will show up in the cell as protein in terms of its DNA that encodes, that is transcribed to RNA, and that RNA encodes a protein. Okay, so when you're predicting genes, we are grabbing this magazine and we are flipping through it, and you're looking where in this magazine are the recipes. And we look for, if this is ab initio, I'm going to look for keywords like ingredients or preparation. So those would be keywords for me to know, oh, here's a recipe. So that's the ab initio. As you know, these would be the sequences that are the start codon, the stop codon, sequences in the promoter, and then there are other things that the program looks for, but let's keep it simple. The homology-based is like this. I have this magazine, and I'm looking for the recipes in that magazine. I want to scan it and just find the where are those genes. And what happens is I am comparing the words from the magazine to words that are in a database of these are recipes. And when we do the BLAST one, when we narrow down the database that you're comparing to RNA or protein, then we know that I'm only comparing to a database of recipes where there's no advertisements included. Okay, so I'm going to open the BLAST, the homology-based exercise, so that you see how that is done, in action at the computer.